So dear students, today we will talk about glycogen, the metabolism of glycogen in the liver as well as in the skeletal muscle. So first of all, we should note that if glycogen form a bond, that is form alpha-1,6 bond and alpha-1,4 bond. So what actually is this? Let's we have a glycogen molecule. So first let's we have a glycogen molecule. This is glucose, this is glucose. A lot of glucose are connected. So between the two glucose molecule, if one we say 1,4, this is between one carbon of the one glucose molecule is connected to the 4 carbon of the other molecule, and this make a straight chain. Straight chain is formed when carbon number 1 of one glucose is connected to the carbon number 4 of the other molecule, then it make a straight chain. But if carbon number 1 of one glucose molecule is connected to the carbon number 6 of the other glucose molecule, then it will form branch. So in this way we say that how straight and branch chain is formed. So in this case, if we see here, this is a straight chain, and that is the bond between the each glucose is 1,4 bond. So this is a bond that is 1,4 glycosidic bond. So this is a glucose molecule in which we have 1,4 glycosidic bond. Now we have another straight chain that come and bind with this, and this bond is now 1,6 glycosidic bond. So this is a 1,6 glycosidic bond, and this bond is actually formed between carbon number 1 of the one glucose molecule and carbon number 6 of the another glucose molecule, and this elongates. So this is the now straight chain. So actually what I want to say that if this is one glucose molecule, this is another glucose molecule, then the bond between this bond is 1 to 4 bond, and this bond, if this is one glucose molecule, this is another glucose molecule, then this bond is a 1 to 6 glycosidic bond. So actually, if you know this, then let's talk about what is actually given here. So first of all, let's discuss from the book. So if we have a skeletal muscle, what actually happen in the skeletal muscle? We know that the skeletal muscle lack glucose 6 phosphatase. So glycogen undergo glycogenolysis. Glycogen is broken down, and the breakdown process of glycogen is called as a glycogenolysis. Glucose 1 phosphate is formed. Okay, you know this. Glucose 1 phosphate is converted into glucose 6 phosphate, which is rapidly metabolized during the exercise. Now the main thing is that here we do not have free glucose molecule because muscle lack glucose 6 phosphatase enzyme. So glucose 6 phosphate enter the glycolysis, and that is used locally during the exercise, and glucose 1 phosphate does not contribute to the blood glucose level. So the muscle glycogen cannot contribute to the blood glucose level. It can contribute to the energy production in this way. So if we talk about the hepatocytes, then in hepatocyte glycogen is stored, and it can be used, and it can contribute to the blood glucose level. So glycogen is stored and undergo glycogenolysis to maintain the blood sugar at appropriate level. So one difference between the skeletal muscle glycogenolysis and hepatocyte glycogenolysis is that liver can hepatocyte glycogenolysis can contribute to blood sugar level, but skeletal muscle glucose cannot contribute to the blood sugar level. So liver has glucose 6 phosphatase, allowing glucose to release blood into the to go into the blood. So this is the main difference between skeletal muscle and hepatocytes. Actually the confusing diagram over here is the main problem for the student. We will discuss this. Let's say we have this diagram, and in this diagram we have a glycogen molecule. So in this glycogen molecule, we know that there is 1,6 glycosidic bond and there is 1,4 glycosidic bond. So actually we have enzyme that is called as a glycogen phosphorylase. So a branch that is called as a enzyme, we have, this is the enzyme that is glycogen phosphorylase. So this is glycogen phosphorylase. This has phosphate groups and give these phosphate groups to this bond. This is 1,4 bond. So when this give phosphate group to this bond, then what actually happen? Let's say this is glycogen phosphorylase enzyme. This glycogen phosphorylase is phosphate groups in it, which it give to 1,4 bond. This is 1,4 glycosidic bond. When it give phosphate group to the 1,4 glycosidic bond, then this bond is broken and glucose 1 phosphate is released. So glucose 1 phosphate is released, and this bond is broken down. In this way, this process continue. It give phosphate to this bond, it give phosphate to this bond, but this process cannot go more than 4 carbon present over here. First, let's say we have 1 carbon, 2 carbon, 1 glucose, 2 glucose, 3 glucose, and 4 glucose. This process continue, and the glycogen phosphorylase break down this bond, but it stop when 4 molecule remain over here. When 4 glucose remain, then this enzyme stop and it does not proceed further. So what actually happen, this release a lot of glucose 1 phosphate. So this is glycogen phosphorylase, this that release a lot of glucose 1 phosphate. Now what will be the final result over here? What will the final effect on this? We have removed these, we have remaining these 4 glucose molecule attached to this branch. Now another enzyme is coming, and what is the name of this enzyme is, that is debranching enzyme. This is an enzyme that is debranching enzyme, and the actual name is 4-alpha gluconotransferase. So this is debranching enzyme. What does this branching enzyme do? This branching enzyme break down this bond and releases these 3 glucose molecule. And what actually this do, this transfer these glucose molecule to the near chain and add over here. So these 3 glucose molecule are added over here. Okay, so what actually happen, this is the branching enzyme that come and break this 1,4 branch bond with glycosidic bond between these glucose molecule. Now these 3 glucose molecule are released. These 3 glucose 1 phosphate molecule are released and added to the nearest straight chain. So this is added, and this new bond, that is 1,4 glycosidic bond is formed, and this straight chain is elongated. Now what will be the final result over here? We have removed these molecule also. Okay, so these molecule are also removed. Now what is actually remaining with us is this glucose molecule. Then another enzyme come and remove this glucose molecule, and that is the actual glucose molecule that is used to increase the blood glucose level. So we have another enzyme, this is another enzyme, that is glucose 6 glucosidase. So glucose 1,6 glucosidase, glucose 1,6 glucosidase. What actually this enzyme do? This is glucose 1,6 glycosidic bond. It break this bond and remove this glucose, and this glucose can be used as a source of glucose in the blood. Then this glucose, this is glucose that go inside the blood vessel. This glucose go inside the blood vessel and increase the blood glucose level. So let's discuss what is actually written inside the book. First of all, we have glucose is converted into glucose 6 phosphate, and then glucose 1 phosphate, and UDP glucose is formed. When UDP glucose is formed, then it gives rise to formation of glycogen. So this is actually glycogenolysis, formation of glycogen. Then we have these glycogen bonds. Now what actually happen, branching will occur at this point, and the branching result in the this structure. So in this structure, if we see, we have 2 types of bond. One is, let's say these are the glucose molecule, these are the glucose molecule. Between these glucose molecule, these are the 1,4 glycosidic bonds, and this is another change of glycogen, and these are the 1,4 glycosidic bond. But if we see, this bond is 1,6 glycosidic bond, this is 1,6 glycosidic bond. Now what actually happen, we have to break down these glucose molecule. So glycogen phosphorylase come, and this is, let's say, glycogen phosphorylase enzyme. It give phosphate group to this branch and cut this, then cut this. Okay, and it remain it leave 4 final glucose molecules. It cannot go ahead of this. So what is the final structure? Then let's play this. So these molecules are removed, and 1, 2, 3, 4 molecule are remain. So 1, 2, 3, 4 molecule present over here are left over here. So let's bring this structure over here. So if we bring this structure over here, we get this structure. In this structure, we have removed 2 molecule of glucose 1 phosphate, and remaining 4 molecule are left. So these molecule, out of these, we should now remove these 3 molecule. Okay, so in another enzyme that we discussed over here is this one, that is now removed. When remove these 3 glucose molecule and head to this here over here, nearest glucose bond. So what will be the result? In the new structure, we will have now these molecule, and we will head this molecule to here. So here, if we see, we have this structure, that is old one, like this one. But to this, we have removed this only remaining one is this glucose molecule, and these 3 molecule came over here. So we finally get this structure. Now we have this glucose molecule. Now we should remove this by another enzyme, that is α-1 glucosidase. Then we remove this glucose by breaking this 1,6 glycosidic bond. This is 1,6 glycosidic bond, and this glucose molecule is released, and finally we get this structure. So this is how what happened actually over here, how the confusing diagram is made over here. In this way, this glucose molecule is removed, and this is used in the glucose level. This is used to increase the blood glucose level. So let's discuss over here glycogen phosphorylase. Glycogen phosphorylase, liberate glucose 1-phosphate residue of branch glycogen until 4 glucose units remain on a branch. So glucose 1-phosphate remove a lot of glucose 1-phosphate, and this will continue until 4 glucose units remain on a branch, as we discussed over here. Here only 4, when we stop when only 4 molecules remain. This is what they are talking about. Then 4-alpha D-gluconotransferase, that is a debranching enzyme, move 3 of the 4 glucose unit from the branch of the to the linear linkage. So then these 3 molecules of the glucose are removed and added over here, and we finally get this result. So this is what they are talking about. Then alpha-1 glycosidase, that is a debranching enzyme, cleave the last residue, liberating a free glucose. Then what actually happen here, enzyme come and release this glucose molecule, and this is released into the blood. Limit dextrin, that is 2 to 4 residue remaining on a branch after glycogen phosphorylase is shortened it. So this is what they are talking about. Here if we see, small amount of glycogen is decreased in the lysosome by alpha-1 glucosidase. So here also if we see in the lysosome also this thing can happen, that small amount of glycogen can also be degraded by the lysosome. So this is the glycogen metabolism.